All right, our speaker's here. Danielle, you want to try sharing your screen?
Yeah. Can you hear me?
Yeah, great.
Perfect. Can you s-
Coming up. Yep.
can you see my screen?
Yeah. Thank you.
Let me
Should
see.
start at your first. Yeah, go to your first. There we go. Thanks.
Yep.
Wait a minute or two here. Wait another minute. People are still joining.
Mhm.
Okay, it's 1 o'clock. We'll go ahead and get started. So it's a pleasure to introduce our speaker today, Dr. Daniel Siddiqui, who is a postdoc fellow in the laboratory of George Kotsakis, who's in our department of oral biology. And we're looking forward to hearing your interesting story about the very important bacterium in the mouth, Veillonella, which is also difficult to say. Thank you, Daniel.
Thank you, yeah, for the introduction. Yeah. So good afternoon, everyone. Yeah, as my name is Daniel Siddiqui, and yeah, I'm a senior postdoc in Dr. Sakis' lab. And today, yeah, I'm going to share with you some of our research regarding Veillonella, specifically how Veillonella catalase allows for its survival and sort of growth with some of the other bugs, such as PG, under titanium-induced oxidative stress, in the context of peri-implant disease. Yeah, beginning with the significance. So dental implants are highly prevalent. There's hundreds of thousands of them being placed annually in the U.S. alone. And in the case of peri-implant health, we have the implants inserted within the bone tissue, and we have this sort of soft tissue seal formation around that that keeps out the microbes in the oral cavity from colonizing the further down the implant surface. However, we can have a condition known as peri-implant mucositis, which occurs in about half of the patients at some point during the implant lifetime, where we have bacterial plaque that sort of accumulates around the implants, and this can lead to sort of this reversible soft tissue inflammation. If this further proceeds, this can lead to what's known as peri-implantitis, where we have destruction of the soft tissue and bone around the dental implant. This occurs in about a quarter of patients with implants. And despite current treatments that are used for peri-implantitis, we see up to a rate as high as 50% or greater recurrence of peri-implantitis despite the treatments. And so, in short, peri-implant disease is becoming increasingly prevalent, and we have no clinically proven effective treatments to date. And so titanium particles are generated from dental implants. They either can come from biocorrosion, or in many cases, when we have the cleaning treatments that are actually used to abrade and remove the biofilm, can actually generate particles which accumulate in the surrounding tissue. And in our lab, we've actually used scanning electron microscopy to study diseased human peri-implant tissue. And as you can see here with these lighter color specks, we've confirmed using EDS elemental analysis that these are indeed titanium particles that are released and accumulate in the diseased peri-implant tissue. And so in short, yes, these implant-derived titanium particles are critical abiotic cofactors that are present in the peri-implant microniche. And so here we propose our model of how titanium particles mediate and microbial dysbiosis in the peri-implant pocket. And so in health, we have commensal early colonizing bacteria that sort of participate in effective immune surveillance. This overall leads to a symbiotic biofilm with relatively high biodiversity. However, in peri-implantitis, when we have titanium particles in higher abundance, this can overall lead to excess oxidative stress condition. And so this oxidative stress actually can change the surrounding biofilm. Specifically, we can have depletion of some of the commensal and health-associated species which are susceptible to oxidative stress, while other bacteria that are more resistant to oxidative stress, such as those that have catalase, can survive. And they can also cross-protect other bacteria that are known to, co-aggregate and grow with them in biofilm. And so this can overall lead to a dysbiotic biofilm with relatively lower biodiversity and higher abundance of some of the pathogens. And so again, in short, our hypothesis is that implant-derived titanium particles disrupt host immune surveillance via oxidative stress, which selectively favors resistant species that ultimately can lead to microbial dysbiosis. So in our previous studies, yes, we have indeed shown that like in peri-implantitis, there is a decrease in the diversity of the community as compared to peri-implant health. And furthermore, in titanium-mediated peri-implantitis, there is an increased an overgrowth of Veillonella as compared to the health condition. As you can see in panel C, on the y-axis, we have the Veillonella abundance, and it inversely correlates with the diversity of the community. And in contrast, the Veillonella abundance positively correlates with the abundance of titanium in the surrounding plaque. And again, in E2, again, amongst all the taxa that are abundant in peri-implantitis, Veillonella are indeed one of the highly abundant species or genre present. And so in short, Veillonella overgrowth positively correlates with titanium particle presence in the peri-implant pocket and inversely correlates with the community diversity. And so a bit more about Veillonella. So Veillonella are gram-negative anaerobes. They're mainly known for coaggregating with and feeding on the lactate that's produced by the Streptococcus species in the oral cavity. And this feature is very important because it reduces the acidity in the localized environment in the biofilm. And this is important for other bacteria that are more sensitive to acidic pH. And so Veillonella can act as a bridging species. So for example, in this figure here, you can see how the Veillonella in blue allow for example, the pathogen PV to not only survive, but thrive and grow in this biofilm with the Streptococci species. And furthermore, some Veillonella species are catalase positive, while others, such as Veillonella parvula, while others are catalase negative, such as the Atypica. And what this means is that when they're exposed to hydrogen peroxide, such as when we do like these in vitro tests, they essentially immediately neutralize and break down the hydrogen peroxide into harmless water and oxygen gas, as compared to the catalase negative species. And so in short, commensal Veillonella species can support growth of other bacteria, especially some pathogens such as PG in oral biofilm. And so a bit more about microbial catalase and how it can actually cross-protect other bacteria under oxidative stress conditions. So in one previous study that was looking into microaerobic condition where we have 5 to 10% oxygen present, they were growing one E. coli strain, which was positive for catalase, in co-culture with another bacteria, the strain P745, that is cannot grow under microaerobic condition. And so when they grew these two bacteria in co-culture on agar, you can see here under oxidative microaerobic condition, you can see here how the E. coli colonies allow for growth of this more sensitive species only around those colonies, and the rest of the agar is sort of clear. And when they deleted the catalase gene from this E. coli strain, you can see how that sensitive bacteria was no longer able to grow on the agar. And similarly, another study looked into how hydrogen peroxide stress affects, in this case, they use fungi, Candida albicans, which is positive for catalase, and they grew it in co-culture with S mutants. And so in co-culture with S mutants under hydrogen peroxide stress, there was relatively large amounts of S mutants that survived and relatively low amounts of S mutants that died. However, when they deleted the catalase gene, there was an increase in the amount of S mutants that died due to the hydrogen peroxide stress. And when they quantified that, you can see here that there was a significant increase in the amount of S mutants that died in co-culture with catalase deficient C. albicans, as well as a significant decrease in the amount of live S mutants. And so, in short, catalase positive microbes can cross-protect oxygen-sensitive species under microaerobic or hydrogen peroxide stress conditions. And so the gap in knowledge that we have here is, does Veillonella catalase, actually cross-protect, provide cross-protection in the peri-implant environment that ultimately can lead to microbial dysbiosis, specifically as opposed to in peri-implant health, where we have higher abundance of commensal species and lower abundance of pathogens. In peri-implantitis, does Veillonella catalase, promote survival of pathogens that associate with it while commensal species are depleted? And so to assess this, we have, for this study, we developed the following aim where we wanted to study and see whether Veillonella catalase is indeed critical to its survival under oxidative stress condition, and whether it can cross-protect the pathogen P. gingivalis. And we hypothesized that catalase positive Veillonella species would resist and cross-protect PG under oxidative stress conditions, thereby promoting microbial dysbiosis. So moving into the methods, so we were performing in vitro tests. So the catalase positive species that we were growing were V. parvula, strain PK1910, as well as wild type SKV38. And we also had a transgenic mutant where the catalase gene from PK1910 was inserted into V. atypica OK5. And then for the catalase negative species, we had, of course, the wild type of V. atypica, which does not have catalase. And we also, with the help of Dr. Carla Cagini, generated a catalase deficient mutant of SKV38. And so we exposed these single species cultures to one of three conditions. Either they were the anaerobic control, or we exposed them to ambient air, so aerobic condition, or we expose them to a sublethal dose of hydrogen peroxide. And afterward, we assessed differential gene expression through RNA-seq of these single species cultures, or we grew these single species or in co-culture with PG and assessed their viability by CFU plating or looking at intracellular stress using flow cytometry. And so we will now proceed with the transcriptomics results. So beginning with, yes, the PK1910, that's catalase positive. As you can see here in this PCA-like plot, so under hydrogen peroxide, Veillonella PK1910 that was exposed to hydrogen peroxide, essentially clusters in one area. And what this graph essentially means is that those that are, the more closer that you cluster together, the closer in sort of like gene expression profile that you have. So you can see here how the H2O2 exposed PK1910 cluster together, and we're distinct from that works the anaerobic control, as well as those exposed to aerobic condition. And likewise, when looking at this sort of cluster heat map of all the genes that were expressed, you can see here how those that are exposed to hydrogen peroxide are quite distinct from that of the control and aerobic condition. So the blue means we have downregulation of gene expression, and then red is upregulation. And as you can see here, it's quite opposite, where we have downregulation here, upregulation here, and so on and so forth. And so in short, PK1910, which is catalase positive, does indeed exhibit distinct differential gene expression under hydrogen peroxide condition versus aerobic and anaerobic condition. And so when we're comparing and looking at the exposure, doing differential gene expression analysis, looking at exposure to hydrogen peroxide versus the anaerobic control, we can see here that many key oxidative stress genes are upregulated. Specifically on, well, here in the y-axis, we have the magnitude of that fold change in gene expression upregulation. And as you can see, catalase was upregulated to the greatest extent, as well as a couple of other genes, DNA protection starvation and superoxide dismutase. And furthermore, this upregulation occurred to a greater extent when exposed to hydrogen peroxide as compared to when exposed to the aerobic condition. And so what this tells us is that, yes, the PK19 catalase positive PK1910 has significantly higher upregulation of key oxidative stress genes under sublethal hydrogen peroxide exposure as compared to exposure to aerobic condition. And now when moving on to the results for the wild type OK5, which is catalase negative and its transgenic mutant, again, when looking at the PCA-like plots, again, those bacteria that were exposed to hydrogen peroxide clustered together and were distinct from those exposed to aerobic condition and control. And again, when you look at the overall heat maps, we can see here how, again, the exposure to H2O2 is quite distinct and different from exposure to aerobic condition and control. And so again, in short, both the wild type and transgenic mutants of OK5 exhibit distinct differential gene expression under exposure to hydrogen peroxide versus aerobic or anaerobic control. And so again, when looking at the key oxidative stress genes that were upregulated in the transgenic mutant, catalase was the highest upregulated gene. And furthermore, we had upregulation of all the other genes. When you compare the wild type in blue versus the transgenic mutant, you can actually see there is a slight sort of decrease in all of these genes. And this is sort of suggesting that the catalase is, doing its role and taking care of a lot of the oxidative stress. And so the other genes don't have to be as highly upregulated. And furthermore, when looking at other genes not that were upregulated, so when we compare the transgenic mutant versus its wild type control, interestingly, we had some other genes such as this hemolysin-like secretion protein, which was significantly upregulated in the transgenic mutant versus the control, as well as hemagglutinin, which is associated with sort of adhesion and aggregation of Veillonella, in addition to some of the other genes associated with lactate metabolism. And furthermore, when comparing the transgenic mutant versus wild type control and PK1910, we can see here that some other genes, such as this P-like sort of complex component protein, was significant or upregulated to a greater extent in the catalase positive species as compared to the catalase negative wild type control. And so, in summary, yes, indeed, catalase positive Veillonella species exhibit higher expression of some adhesion aggregation genes under hydrogen peroxide stress as compared to the catalase negative species. And so now I'm going to move on to the in vitro results for the viability of intracellular stress. So here in this plot, we have on the y-axis the bacterial counts, so enumerating basically the viability of the bacteria. And so the blue bars are the control untreated condition, and then the red bars denote exposure to hydrogen peroxide. For these tests, I actually exposed them to bursts of hydrogen peroxide. I did 4 doses with 50-minute intervals. And so what happened was, under this condition, the catalase negative OK5 did not survive, essentially was depleted below its limit of detection, while the transgenic mutant was essentially completely survived to a similar extent to the PK1910 control. And likewise, when you looked at the intracellular stress levels, the wild type catalase negative species had significantly higher, an order of magnitude higher intracellular stress level as compared to the transgenic mutant and PK1910 control. And so this shows us that the insertion of the PK1910 catalase gene into OK5 rescued its survival and reduced intracellular stress under this sort of burst of H2O2. And then likewise, when with another strain of Veillonella parvula, SKB38, which is positive for catalase, when the catalase gene was deleted, we saw that there was a decrease in its viability under exposure to hydrogen peroxide stress as compared to the its control and the wild type control. Furthermore, under the intracellular stress, we again see that there is an increase in intracellular stress in the catalase deficient mutant as compared to the catalase positive control. And so this also shows us that deletion of the catalase gene impairs the survival of SKB38 and increases intracellular stress under bursts of hydrogen peroxide. Now moving on into the cross-protection assay. So in co-culture of the SKB38, the wild type or catalase deficient mutant with PG, so when looking at the CFU of Veillonella, you can see here how under exposure to hydrogen peroxide, it essentially completely survived, while the catalase deficient mutant had a orders of magnitude decrease in its viability. And likewise, PG, while it did decrease, was able to survive with the catalase positive wild type, while in the catalase deficient mutant, it was essentially eliminated below the limit of detection. And when we looked again at the intracellular stress levels for the total sort of biofilm, both Veillonella and PG, we can see here that there was an increase in PG and Veillonella and PG that were grown in co-culture under oxidative stress when you don't have catalase present as compared to the catalase positive control. And likewise, when we were looking at just stating for PG, we again see that the PG intracellular stress was higher in co-culture with the catalase deficient mutant under hydrogen peroxide stress as compared to the control group and control condition. And so in short, catalase positive SKB3 cross protects PG and reduces intracellular stress in co-culture to a greater extent as compared to the catalase deficient mutant under hydrogen peroxide stress. Furthermore, we also actually stained the bacteria. We formed some fish staining. And so on the left-hand side with the catalase positive SKV38, the Veillonella is in purple and we have, or magenta, and we have PG in this fluorescent green. And under, with no hydrogen peroxide stress, both the Veillonella and PG aggregate and cluster together, and we have abundance of both Veillonella and PG. And immediately after the exposure to bursts of hydrogen peroxide, the Veillonella mostly survives and it can protect some of the PG, as opposed to the catalase deficient mutant, which again, under the untreated control condition, we have both of them present, but when we expose them to bursts of hydrogen peroxide, there is a decrease in the amount of both the Veillonella as well as the PG. And so again, we see here how the catalase positive Veillonella can cross-protect PG in dual species biofilm as compared to the catalase deficient mutant under a burst of hydrogen peroxide. Furthermore, we also performed a sort of a Transwell cross-protection assay experiment. And so what I did here was seed Veillonella in the bottom of this bottom chamber of this 24-well plate, and then in the upper chamber, I seeded PG. And in this first experiment, I dosed the hydrogen peroxide in the Veillonella chamber. So essentially, the H2O2 is seeing the Veillonella first before the PG. And so under these conditions, the Veillonella, again, the wild type catalase positive control did not experience any significant decrease in viability, while the catalase deficient mutant had a decrease in its viability, viability. And likewise, we see the same trend in terms of PG survival. And so again, in short, exposure to hydroperoxide, well, firstly to the Veillonella before PG resulted in protection of PG. However, when I dosed the hydroperoxide first in the PG well, so it's the H2O2 seeing the PG or PG seeing H2O2 before it reaches to the Veillonella. Both Veillonella, wild type and the mutant survived, while the PG was eliminated in both in co-culture with the catalase positive and catalase deficient mutant. And so exposure to H2O2 to PG before the Veillonella parvula resulted in the death of PG. And so in summary, catalase positive Veillonella significantly upregulate oxidative stress genes, as well as other genes associated with adhesion aggregation under H2O2 condition. And indeed, the Veillonella catalase is critical to its survival under burst of H2O2. In our study, in terms of, we assessed that with CFU viability counts, as well as intracellular stress measurements. And furthermore, catalase positive Veillonella are able to cross-protect PG in co-culture to a greater extent than catalase deficient Veillonella species when exposed to H2O2. And lastly, this Veillonella cross-protection is dependent on, of course, catalase as well as the proximity to PG. So in overall summary, the catalase positive Veillonella species are indeed an important accessory pathogen with the potential to mediate dysbiosis in the periimplant microniche, and this requires further investigation. And so some of the ongoing and future studies that are currently taking place, in one study, we are performing we have an oral mucosal abscess model, and we are injecting either the catalase positive or catalase negative Veillonella species, either the single species by themselves or with titanium particles into the oral mucosa of the mice. And we are to generate an abscess, and then we are studying and looking at the viability of the recovered bacteria in the abscess by CFU plating, as well as looking at catalase expression using qRT-PCR, as well as measuring the abscess size. And so we actually do have some results here. So in a dose-dependent manner, we can see here actually how the Veillonella parvula results in a sort of a chronic abscess that forms that is not resolved, while in the case of the catalase negative VA typica, there is resolution of that. And here, when we plot the surface area of the abscess, we can see here how, given enough catalase positive Veillonella parvula, we have a persistent chronic abscess that's relatively large in size that persists up to 2 weeks, while the catalase deficient Veillonella atypica, when inoculated to a similar concentration as that of the VP, we can see here how it's not able to form a chronic abscess and it resolves. And so we're also repeating these experiments with titanium particles, and we're to assess the capacity of Veillonella catalase to resist titanium-induced oxidative burst in vivo. Furthermore, we also are assessing how Veillonella sort of modulates the community composition of some periimplant-derived ecological biofilms. And so again, we're using a transwell sort of system. In the upper chamber, we have sort of macrophages that we are stimulating with titanium particles to secrete excess oxidative burst, mainly hydrogen peroxide. And in the bottom chamber, we have these biofilms that are derived from periimplant plaque that are grown on titanium and then exposed, of course, to these titanium-induced oxidative bursts. And what we're hoping to study and see is how, when we have Veillonella parvula, again, catalase positive present in these biofilms, we're expecting to see that this will result in a dysbiotic biofilm, where we're going to, again, have cross-protection of the pathogens and late colonizers, which is going to lead to lower diversity. However, when we don't have Veillonella parvula present, we're expecting to see depletion of those pathogens due to the lack of cross-protection present. And then when we spike these communities with Veillonella parvula, we're expecting to see a dysbiotic biofilm forming. And so in short, with this sort of model, we're expecting to study and evaluate the impact of Veillonella catalase on polymicrobial dysbiosis under titanium-mediated oxidative stress. And so, yeah, lastly, I'd like to, of course, acknowledge the support of my mentor, Dr. Kotsakis, as well as the rest of the Kotsakis Lab members, the current ones and previous ones, as well as all our collaborators and our funding sources. And so with that, I conclude. Thank you, and we'll take any questions.
Thank you very much. Very nice presentation. Anybody have any questions for Dr. Siddiqui? You just put your hand up or put a question in the chat box if you have. See. Anybody have any questions? I see a lot of clapping. Can't hear it, but I see it. Okay, Pia, you had a question? Please, you can turn your microphone on. I saw a hand up. I don't know if somebody have a question, Pia? Or was that just a accident? Let's see. Okay, she meant to clap. Yeah, okay. No problem. Okay, well, if nobody has any questions, oh wait, let me just, is there a chat box? Okay, yeah. Okay, if nobody has any questions, thank you, Daniel, very much. Very nice presentation. We look forward to hearing your follow-up with your studies that you've already started, your ongoing studies and future studies. So thank you, everybody. Have a great weekend.
Yep. Thank you. Yeah.
Take care.
Thank you. Great
Thank you.
presentation.
Yeah, very
Thank
nice.
you. Yeah. Yeah. Bye.